Thank you, Lucy, for that very nice summary. I'm going to talk about what causes ALS and try and put things into perspective. The reason it matters to know what causes ALS is because if we're trying to design a cure, we need to understand the nature of the disease to make a rational design of new drugs so that they can be targeted correctly. This is my head reconstructed in 3D from MRI scans. And one of the key problems in ALS is understanding why this particular set of neurons, just here in the motor cortex, and down here in the bulbar region of the brainstem and also in the spinal cord, why the neurodegeneration is restricted to those areas. Why doesn't it affect all nerves? This is a key fundamental problem in ALS. The advancer isn't working.
Yes, it does eventually.
Does it? Okay. That's it. Thank you. So this is a description of what happens to all of us. We're conceived, we're born, at some point we develop a severe illness, in this case a motor neuron disease, ALS, and then that disease carries us off. The causes of that final illness act somewhere. Thank you. The causes of that final illness act somewhere between conception and the onset of that illness. And there could be one cause, multiple causes, or they could be acting at different times, or maybe acting as triggers to some underlying cause. But we don't meet people at that point. We meet them after diagnosis. And so we're trying to find out what causes the condition, the things that have happened before diagnosis, from people after they're diagnosed. And that poses particular problems. So how can we understand the causes of ALS? Well, one way is to think of it as we think of most conditions, really as a combination of genes, environment, and some other things that we don't fully understand, which we can call random effects, because they'll appear to have randomly caused the condition. And of those three things, it's very difficult to understand random effects with science. Environmental studies are very difficult because we're meeting people after diagnosis. And the only way you can really get at environmental studies is through questionnaires at the moment. And trying to ask somebody about something that may have happened in their very early life, for example, how well did you sleep when you were 3 years old? Or did you used to eat apple cores as well as the whole apple or just the outside of the apple? There's millions and millions of things you could ask, and most of them are impossible to recall. So tackling environmental problems is actually very challenging. There are ways around that, which I can discuss later. Parental genes, understanding the genetic causes of the condition, is a tractable problem. It's technologically difficult and it's very expensive, but it is something that is at least a finite search space that could have an answer. So that's where we tend to focus our resources. This is one way you can think of how ALS happens, that we're born with some kind of genetic load. At some point, as time goes on, we age, we develop cell damage, and we also have environmental exposures. But unless we reach some critical burden of those disease-causing factors, we don't develop disease. So that's gene, time, and environment, those three components. For some people, they do develop enough of that burden that they will then develop, in this case, ALS, and that's, we think, a self-perpetuating process that then progresses. And in some people, it seems that the genetic load is bigger, and in other people, they may have lots of small genetic risk factors and more environmental risk. And in many people, probably, there's an underlying genetic risk, but actually that genetic risk only matters in the particular context of the things they've been exposed to in their lives. So if you're not exposed to those things, you won't get the disease, or if you don't have that genetic component, but you are exposed to those things, you won't get ALS. So you probably need both. So how can ALS be both environmental and genetic? A big problem to understand is if you carry a disease gene, how come it only affects you when you're 50 or 60 or 70? You've had this since conception. This graph you can see here is a risk curve. It's how likely you are to develop ALS as an individual. So you've probably heard the incidence of ALS is 1 to 2 per 100,000 person years, or the prevalence is 5 per 100,000 population. Those are very difficult units for a normal person to think in. I don't think in 100,000 person years units. But if you think, what's your risk as an individual of developing ALS? It's 1 in 300. That means of every 300 people you meet, one of them in their lifetime will develop ALS. So if you came over on a plane to Orlando, if it was a jumbo jet like I did, there are probably about 600 people on that plane. So 2 of those people will develop ALS in their lifetime. And you can see from this risk curve that the risk is greater in males than females, and the risk increases with age. As far as we know, it just carries on going up. So one way we could explain this is if ALS is a multi-step process. And there's some very complex maths on this, which I'm happy to go through with people individually if they want. But the bottom line is that if ALS is a multi-step process, then we could plot a log of the incidence with a log of the age. So how frequent is it at each age? And if that's a straight line, then that suggests it's a multi-step process. So we can test that with population registers, which are registers that capture every single person who develops ALS in a particular time frame in a defined geographical area. And if you do that, if it is a straight line, then the slope of that line gives you a clue as to how many steps are involved in ALS. So this is the result from the Southeast ALS register in the UK. So you can see it looks like a nice straight line. The R-squared value there is 0.95. R-squared is a measure of how straight that line is. How straight do those dots line up? So the closer it is to 1, the better. So that's 95%. That's extremely straight. This is the result from a Netherlands population register. R-squared is 0.99. And from an Italian register, 0.95. An Irish register, 0.99. Scottish register, 0.97. So you can see these 5 population registers, all independent. They're all European population registers, but they're all independent registers. They all suggest that ALS is a multi-step process. And what's interesting is they all give, there's the overall score, 0.99, they all give the same slope, which is about 5. And if you add 1 to that number, that gives you the number of steps that ALS is on average, how many it takes. So that's a 6-step process. So what that means is these gene, time, environment exposures that are leading you to develop the critical burden of disease, there's on average 6 steps needed to reach that threshold. And that explains a lot of things about ALS. For example, it explains that genes and environment are needed because some of the steps may be genetic, some of them may be environmental. It explains why it comes on in later life, even if someone carries an ALS mutation, because you have to have lived long enough to have been exposed to the other things that will carry out the other steps. It also explains why one gene can result in different diseases. For example, why can expansion in the C9orf72 gene cause ALS or frontotemporal dementia or both or nothing? And it's because it depends on exactly which steps you take. If you take steps A, B, and C, and D, E, F, you may have a different outcome from if you took steps Z, Y, X, and D, E, and F. It also explains why some people don't develop ALS even if they carry an ALS gene variant, because they won't have necessarily gone through all the steps. And it explains why ALS could have many causes. Of course, this assumes that ALS is one condition, and we think it may well not be one condition. But how do we find the causes of ALS? Well, it's impossible really in a relatively rare condition to do this as a single lab on your own. You have to work together. And this is something that's happening much, much more now. Mainly because of the internet and because of connectivity, and also because the ALS research community is very tightly knit. So we have international consortia. These are just 3 of the ones I'm involved in. I'm involved in many more. The Strength Consortium is one I lead. Its aim is to look for subgroups of people with ALS. So this personalized medicine that Lucy was talking about earlier. Because if we can find the subtypes of ALS, if we can show that ALS isn't really one big blob of a condition, but actually there's lots of different types, it may be that we should target clinical trials at one type, or that we should analyze our clinical trials taking those types into account, because maybe it works in one group and not in another. It'd be like if you gave penicillin to everybody with a chest infection, but only some people got better. You wouldn't necessarily know that if that subgroup that was susceptible to being cured by penicillin was only a small group. You'd have to know, okay, they've got the bacterium that's susceptible to penicillin, that's why they got better. We're also finding the genetic causes of ALS. That's very difficult on your own. Again, you have to have tens of thousands of samples because you're looking for small common variations or very rare variations that have a big effect. Finding markers for ALS also needs you to have large groups of people because we need large numbers of sample. And clinical trials require large numbers of patients to take part so that we can understand the outcome. So with all of this, we get better understanding and therefore a better chance of making a proper treatment. So how can we think of ALS in different ways? How can we work out what these different groups are? So one way is to think like a neurologist. When I see people in clinic, I say, well, this person has typical ALS. This person had a floppy weakness affecting both arms. It's quite symmetrical. I'm going to call that flail arm syndrome. This person has swallowing difficulty. I'm going to call that bulbar onset ALS. Each neurologist has their own particular classification. This one's mine. And what you can see on these graphs is what's called a Kaplan-Meier curve. And that's a curve of how long people live with the condition. The way the curve works is everybody starts off alive, so that's at the 1 level. It's the proportion of people left alive as time goes on the x-axis. So that means that the more horizontal the line, or the more to the right the line is, the better the survival. And my classification is the one on the left where it says neurologists, classification predict survival. So I just took the information I had available to me in clinic on the very first day I saw somebody, and then many years later we went back and looked at the survival of those people. And you can see a proportion of people actually live far more than 10 years. But for most people, the situation is not that optimistic. You can also see that some groups of people do quite well. So that purple line, which is the flail leg syndrome, people with that condition have a 50-50 chance of surviving about 6 or 7 years. Now, we gave the same information to a computer and said, can you classify, using this same information but with no clues, can you classify this information into groups? And it came out with 5 clusters. And then we looked at the survival, which we hadn't given the computer. We looked at the survival of those 5 clusters. And what you can see is the computer pulled out from information at the first visit this yellow horizontal line. That's 3 or 4 people who didn't die during many, many years, during 20 or 30 years. It also pulled out this group with the reddish line, which is people with an average survival of about 15 years. So to the computer, it was obvious that there were these subgroups that have much better survival, but these aren't apparent to a neurologist. So understanding what are those subgroups is very important. This is an example of a genetic study. This is a comparison of 17 million genetic variants in 13,000 people with ALS and 25,000 controls. So at each of those 17 million genetic variants, we've asked the question, does that variant occur more frequently in the people with ALS than the controls? And then we've plotted a dot on this graph. So 17 million dots on this graph. Every time it goes above this very faint horizontal dotted line here, that means there is a significant difference between the 2 groups. And you can see this large skyscraper in the middle. This is called a Manhattan plot because it looks like skyscrapers. There's not much of a Manhattan skyline at the moment for ALS. But what you can see here is this is the C9orf72 gene signal. But you can see there's actually about 8 signals there. So we can use that to try and dissect out what's the jigsaw puzzle, what are the proteins, what's the pathway that's causing ALS. We're also taking part in Project MIND, funded by the Motor Neurone Disease Association in the UK. That is an international whole genome sequencing study aiming to sequence 22,500 people's genomes. To put that in context, to sequence one person's genome cost $3 billion and took 16 years the very first time it was done. It now can be done in about a week for about $1,000. So the technology has really rapidly advanced. And the main problem actually is handling the data. Each person's data comes back as 1 terabyte of data. So we're going to end up with 22,500 hard drives before they're even backed up, before they're plugged into computers and analyzed. It's actually the data analysis that's the challenge here. But what's very good about this data is it has multiple layers of information. We have the clinical pattern for that particular person. We often have other information that went with that person. They may have filled out an environmental questionnaire, or they may have also given us a sample for what's called epigenetic studies or for RNA studies. So we have multiple levels of scientific information on each sample. And genetic discovery is doubling every 4 years. So what we're hoping is by understanding the genetic risks, we can then put together the jigsaw puzzle and understand what causes ALS. That doesn't mean that all ALS is genetic. What it means is there is a genetic contribution. And that means that, just as I showed with the multi-step model, you need more than the genes, you need the environment as well. So just to conclude, international collaborations are vital for us to make progress. Technology is only part of the answer. We have to have patient participation, and we also have to have creative ways of answering the scientific questions, such as using computers or using clues from the cancer world like we did the multi-step model. Thank you very much. But I'd also add that one of the ways we've tried to find new drugs is to test them in model mice. Those are mice that develop motor neurone disease, or ALS. And the mouse that's been used traditionally is one that has a mutation in a gene called SOD1. Now, that's the mouse that we can cure quite well with drugs that it doesn't seem to translate into people. But if we look at the pathology of people with motor neurone disease, if you look at the spinal cords of people with ALS, they don't usually have any pathology that involves SOD1. they've got pathology that involves another protein called TDP43. And about 2% of people have pathology in SOD1, and the rest have a problem with TDP43. So perhaps what we really need to do is develop a better mouse model, and if we can cure that mouse model, it might translate better into people. We've probably been looking in the wrong direction all these years. Another reason is that it's this stratifying, this precision medicine problem. We're looking at ALS as a whole, as a blob, as a single disease, but actually it's probably lots of different conditions, and we need to be a bit more nuanced about how we test our treatments. And the third thing I'd really like to say is the thing that's really surprised me, I appreciate those questions, the thing that's really surprised me is why we haven't taken Riluzole and tried to make a better version of it. Because with other drugs, with other diseases, as soon as a drug is successful, you get lots of Me Too drugs. If you take migraine, people, the first drug that came out was called Imigran or sumatriptan. Immediately there was rizatriptan, zolmitriptan, there were a whole lot of other triptans that were invented. Why haven't we got other Riluzoles? That's what I'd really like to see.
When the person has extreme stress symptoms, and very often they don't get helped. They don't have rehab, they don't have psychiatric help and psychology. Basically, I've given some example. The kids in college, my son's college, they come to me and say, I'm stressed out, I can't sleep. Can I take a drug? I said, no. You don't do that, and get some good sleep. But they don't have anyone advise them to get good sleep. So they stress out. They tell me, my biological clock is not 24 hours, it's 36 hours. I said, no, this has to be wrong. So a lot of people are poorly advised. So that's what I'm getting at.
So your question really is, so you're really asking, are people, when they're newly diagnosed, how do we help them cope with that diagnosis?
No. When a person has symptom onset, can we do something at that point to quickly bring down their stress level by good sleeping help?
I think the problem is we see people at too late a stage to really be able to help. By the time someone comes to a doctor, a neurologist in the UK, and this is true worldwide really, about a year has passed since their first symptoms.
Right.
So it's too difficult to do that.
The key is, can we do something when they just had a symptom?
Yeah. The difficulty is knowing that those symptoms are from ALS and not from something else.
It doesn't matter. If somebody is stressed out, figure out how to help. That's my question.
People go to their local centre, they donate their clinical data, they answer environmental questionnaires, they may give a tissue sample or a DNA sample. And really what you're asking about is data sharing, because the issue comes down to that sample is analysed by the relevant expert in the field. So, for example, if it needs a genetic analysis, there's only 2 labs that do that to a high level in Europe, and it'll go to one of those, and then they share the data with everybody, not only the people who donated not only the lab that donated the sample or the clinic that donated the sample, but it's available to everybody within the European consortium. And actually, we also make that data available to people in the US and Australia, provided that consent has been given for that. So I think it's a very simple system. You basically have donation at one centre, and then the data is shared as it needs to go. The sample can be sent out to where it needs to go as well for other things. So, for example, if you have a blood sample, that can be used for RNA, that'll go to the RNA centre. A bit of it will go to the DNA centre, and a bit of it will go for methylation analysis in an area that does, in a centre that does epigenetics. Which I think actually happens in the US and in Europe, is we have a single identifier that's anonymised, that you generate the same ID from the same input information, so that we know if there are duplicate samples. So the way we do it is very similar to the way that it's done in the States. So you give the first name, the last name. We also have to account for different spellings, and we have a single alphabet that will do it. Date of birth and year of onset is what we use, and then that's used to encrypt to generate what's called a hash. And that's a very, very long string of letters and numbers. That's a unique ID for that patient. So if they go somewhere else, it'll generate the same ID, and then in the database they'll be matched up as the same person.
And can you use those data that you take from the patients in Europe for epidemiology studies too?
Yes, we do. So we have, so the data I showed in my talk, that was from 5 different population registers. Those are some of the epidemiology studies that are done. We've got tens of thousands of people in epidemiology studies. And I'd like to add that we've done a study in which we've examined exactly that question. And we found that having a purpose in life, which could be anything from having a job or having a child to look after, any purpose in life, has an effect about as big as the effect of realism. I would add that it's really the fact that we now have the ability to do massive, large-scale collaborations that are truly international. Tens of thousands of people participating, many centres from lots of different countries, and we can study different levels of information from the same person. That really makes a much, much more powerful data set for analysis. I think that's really going to lead us forward.
I'm Lori Fieldman from the Les Turner Foundation in the Chicago area, and I wanted to go back to the positive attitude question. I have the privilege of counseling patients and their families in home, so I work more with the quality of life than the quantity of life your research is trying to increase. But I was wondering, did you say that you had a study, and can you say a little bit more about that in terms of purpose? Because I talk with families about that at least 2 or 3 times a week.
I'm happy to send you the paper. It was.
I would love to see that.
It was basically a qualitative study, so it was a semi-structured interview of a set of patients and carers. And it was looking at multiple different aspects, and the thing that really found out was having a purpose in life. But that could be, the purpose was undefined, so it could be anything like a hobby, a family member who cared about very strongly, etc.
Yeah, we see that all we're fortunate enough to have a home team, and we all see that. Yeah, so I would love to see that. Thank you.